How do cells control what enters and exits their cytoplasm? To answer a question like this, we need to first know how molecules move on their own, and then examine the ways membranes influence that movement. First of all, how do molecules move? Specifically, how do they move in solutions, such as the cytoplasm of our cells or the extracellular matrix outside of our cells? In any solution, there are two categories of chemicals, solutes and solvents. Solvents are the common molecules throughout the entire solution, into which the solutes are placed. When solutes are placed into a solvent, the molecules of solute tend to spread out from their area of high concentration down to their area of low concentration, while the solvent does the opposite, spreading from its area of high concentration to its area of low concentration, until these molecules are evenly distributed throughout the solution. In biological solutions, the solvent is almost always water, while solutes could be many things. For example, glucose, amino acids, ions, all of these are solutes floating in and around our cells. When molecules are on their own, they follow this pattern of diffusing down their own concentration gradients, the solutes away from each other and the solvents away from each other, until molecules are evenly distributed. That's a little bit different when cells are involved. This is because most cells are surrounded by a plasma membrane or cell membrane such as the one shown here. Cell membranes are made of many kinds of molecules, and those molecules work in concert to control what can enter and what can exit. Let's look at the three major ways a membrane can control what passes across. First off, there is simple diffusion or diffusion, in which molecules make their way across the membrane all by themselves from high concentration in one area to low concentration in the other. Sometimes molecules can't fit across the membrane by simple diffusion, though, so instead they need facilitated diffusion. French and Spanish speakers know that facile means easy, and so facilitated diffusion is the making of diffusion easier. So you can see these molecules might not be able to get across themselves, but with the help of this protein channel that's embedded in the membrane, now they have a little tunnel that they can pass through. So diffusion of these molecules was made easy thanks to the cell doing facilitated diffusion. Both these two types of transport are called passive transport. So they don't require any energy, and as long as the channel is there, these molecules can move by themselves. Even without a channel, these molecules can move by themselves. They're just moving right down their concentration gradient. Contrast that with the third type, active transport. You can see there's a difference here because this molecule is less concentrated out here and more concentrated in here. Sometimes the cell wants to fight against diffusion and keep valuable molecules inside, so while these molecules might want to spread out and move in that direction, the cell is fighting that and saying, no, get right back in that cell, I want to keep you here in a high concentration. And in order to do that, the cell runs a process called active transport. Okay, let's look at each of these three types of transport in a little bit more detail. We'll start first with simple diffusion, where molecules just pass across the membrane themselves. And to understand this, we must remind ourselves about the properties of these phospholipids that make up the membrane in this region. Each phospholipid in a membrane has a hydrophilic head and hydrophobic tails. Hydrophilic means that the head is charged and that water could hydrogen bond with it, while the tails have no charge, and thus the water would not be able to adhere to it at all. Therefore, when many phospholipids hang out in solution, they tend to form structures such as this, with the hydrophilic heads around the outside interacting with the surrounding water, while the tails face away from it in the center. If phospholipids are surrounded by water on two sides, then they form a bilayer, bi meaning two, with the hydrophilic heads of each layer facing outward towards the water, and the tails forming a hydrophobic layer in the middle. If this structure gets folded around on itself, it can make the membrane that goes around the outside of a cell, with water inside, water outside, and the hydrophobic tails safely tucked away in the middle of that membrane. So how does the membrane control what can pass in and out of the cell? The phospholipids are selective. Molecules can move across the membrane by simple diffusion if they are small enough. Like these blue solutes seem small enough to slip between the gaps in the phospholipids, but these orange ones are too big. So in this case, the phospholipids are selecting the small molecules only to pass across. Not all molecules get across just because they're small, however. This molecule right here is somewhat sizable, but it can pass across. What is it? It's called testosterone, and it's also a lipid. Lipids, as you know, are all hydrophobic, so even though there's a hydrophobic barrier through the middle of the membrane, the testosterone molecule can still move through because it is also hydrophobic. However, if the molecules are too large, or if they are hydrophilic, typophil, they need to have their diffusion facilitated or made easier. You can see in this diagram we've got solutes that are highly concentrated up here outside the cell and slightly less concentrated inside the cell, so these molecules would have a tendency to diffuse down their concentration gradient into the cell. However, they're too big or too hydrophilic to make it down this way, so they need a channel to get through, and that's exactly what the cell has put inside its membrane, a protein channel. Protein channels simply make a little tunnel or little passageway so that those solutes can move down their concentration gradient as they would normally. However, the cell needs to do a lot more work if it wants to push molecules against their concentration gradient. Take, for example, in this picture, now the solutes are more concentrated inside the cell. Look at this, 1, 2, 3, 4, 5 solutes down here, whereas there's only 3 outside. And yet perhaps this solute is very valuable to the cell, and so the cell wants to hold on to it as much as possible. In this scenario, the cell pumps substances against their concentration gradient by active transport. Active transport requires a different kind of protein called a protein pump. Protein pumps change their shape, so they might start open in this direction so that a solute can go in, and then when they're ready to push the molecule through, they change so that the other end is open, and the solute can continue its journey down into the cell. This is active transport rather than passive transport because it doesn't happen on its own. The molecules, if they had their druthers, would be moving from a high concentration inside the cell to a low concentration outside. Yet the cell is fighting that tendency, so it needs to do so by active transport using a pump, and that requires energy. It won't happen on its own. What's the source of energy? You guessed it. An ATP molecule is required each time the pump pushes a solute. To recap, molecules tend to spread out from areas of high concentration to areas of low concentration by diffusion. Cells can either use that tendency, help that tendency, or fight that tendency in order to control what goes in and out of cells. If molecules are small or hydrophobic, they can move across the membrane through the phospholipids by simple diffusion. If the molecules are large or hydrophilic, they can move through, but only with the help of a protein channel. That's called facilitated diffusion. And if the cell wants to push molecules against their concentration gradient from low concentration to high concentration, then it must use a protein pump to push it and ATP to power the pump. And that's called active transport.